So welcome, everyone. It's really, really great to be in Boston, Rachel's hometown. I've never been here before. I was told it's really cold and there's a lot of snow, and by Jim Breyer for about 10 years. And there's no snow, and it's really lovely. So the band that we kicked off the session with is, of course, the Pixies, who influenced an enormous amount of bands that certainly I grew up with. Over the next session, Lakmal and I are going to give you a little bit of an insight into where is our mind at Oxford Nanopore in the Innovation Hub, and hopefully give you a little bit of a sense of what might be coming. We will be making some forward-looking statements, and of course this is the first most exciting slide that everyone should read in great detail. But thank you so much to the Nanopore community. It's been 10 years since we've been doing these customer events. It's always really, really brilliant to hear your feedback, to interact with you at the live lounges, at the evenings that we have. And it's really, really special, and we hope that you enjoy it as much as we do. So just to kickstart with the platform, many of you know this, but in case there's anyone new in the audience, we have the foundation of our platform is really that label-free, single molecule scaled sensing technology. So we place at the heart of everything that we do are our motor enzymes, our nanopore proteins, or all of the sequencing chemistries around them, and we scale these through from personal sequencers to really high throughput devices. The fact that we sequence native molecules enables us to really get into a multiomic paradigm. And the talk earlier today, if you watched it by Phil in our applications team, really showed the power that multiomics data can deliver and really, really drives all of the really exciting publications that we see from the phenomenal community that we have. So all of these richer insights, these really fast time to results, this accessibility, affordability, the fact that a 16-year-old was up on stage making all of us look really bad because he was brilliant at delivering his talk. All of that is what's really driving the platform adoption. So the platform today, where it is today, we're running our E8 series of motors, our R10 series of pores. We're sequencing DNA and cDNA at 400 bases per second. We have a high accuracy base caller. It keeps up on all of your devices. It delivers over Q20 performance, and that is all released in Minnow. Our algorithm teams have been working tirelessly. I was about to say effortlessly. It's definitely not effortlessly. It's tirelessly to continue to push the boundaries of accuracy, and our SUP base caller is Q26. We've done an enormous amount of change. It's required a lot of change to get to this position. We really want to thank the community for all of the patience that you have with us as we introduce new pores, as we introduce new enzymes. But what we've managed to do is get into a really simple, much simpler product space for 2025. With our R10 on Flongol, R10 and RNA on Minine. We also have Q-line for Minine because we know that there's a lot of applied market users for it. For that, we have R10 and RNA on PromethION, and all of our Kit 14 products are all now up to date. And because we've been pushing all of the boundaries of accuracies on our simplex data, we are now able to retire the high duplex, and we're working on something very exciting for anyone who's doing hemimethylation, because that is a really, really interesting thing that's come out of the high duplex early access. So completing our lineup as we get ready to move into 2025, of course, this year we've launched P2I. We're getting into the final throes of getting the Mark 1D out, so it's been out with beta testers. It's moving into early access in Q4. Feedback so far has been brilliant, so it is our USB-C upgraded Minine. It's definitely got improved thermal control. Someone's been putting it in fridges and incubators to really kind of make sure that we've done our homework and it's working. Installation is will be much simplified, and of course there's a future compatibility with the iPad that's been out on the live lounge today. So whilst we continue to innovate in the life sciences, we do know that our applied product users really want a lockdown product. So the Q-line GridION is available now. and that is feature frozen. It's a much, much streamlined user interface. It's 21 CFR Part 11 compliant, R9 chemistry, and there's also a biopharma version that we're working with our biopharma customers on that can do RNA QC and plasmid workflows all pre-programmed. In 2025, the teams are going to be working on delivering PromethION and getting the GridION all upgraded to R10 and RNA. If anyone is really trying to get ahead of the game, here's a little insider tip. The likelihood is that the piece of software that's going to go into Q-Line is heavily based on Minnow6. So if you want to get going, you can start now Minnow6 for your PromethION and GridION R10s, and that is what is going to be appearing. So moving on to pushing the performance. Machine learning has been something we've been doing for years and years. You've heard a lot of talks over the years about how we really drive all of our improvements through base calling, through algorithms, and the team continue to do this. But they've also started to look at what is possible beyond base calling into the rest of the informatics pipelines. So we have the core tools, you've got variant detection, you've got the complete pipeline workflows, and we're really now starting to explore how we can optimize all of these for Nanopore. So on base calling, we showed it at London Calling. We showed our transformer models. The teams have just pushed out Dorado V0.8. That includes significant speed increases for the transformer models in that. We showed Dorado at London Calling. So this is a polishing step. It delivers Q40 simplex pre-assembly reads. Again, we've done significant stability improvements to this tool in Dorado since London Calling. It's all based on Hero, and that is available as of 20 minutes ago. And the team have also been looking at how to improve the training sets, making sure that we've got a lot of diverse contexts in there to keep driving our platform performance up. And so this is an example of what we can do by improving training on bacterial data sets. And you can see here that on the bacteria, the very, very vast majority is Q60. But for our team, that is not enough. We still need to drive further. So we're now looking at Medaka. For anyone who may remember Medaka, it's a tool that we have had for a while, and the machine learning team are now looking at optimizations of this. And what they've managed to do by adding improvements into the polishing is about halve the number of genomes that weren't quite getting to Q60. So keep pushing that envelope forward. We've also been working a lot with our plasmid partners and putting a lot of effort into ensuring that we're far, far better at plasmid calling in difficult regions, in regions that have got hard to reach areas. And again, the team have done a significant lift there and really, really improved the performance of our plasmid product. How to improve indels? Well, you will hear, of course, Lakmal, who's our nanopore guru. Nanopores in chemistry is certainly one of the ways. But how about pushing much, much harder on what Medaka could do on indels? And the team are exploring this. Not quite ready yet for release, but this is in development. So they're looking at additional sources of information, like Q-scores, like dwell times, that we could be using to drive the performance, our indel performance up. And the initial results are incredibly promising. And finally, onto variant calling, working with the authors of CLAIRE 3, we're really pushing up the performance of the variant caller to get increased, to get a reduction in errors at 30x coverage. So in summary, where the platform is today, over Q20 simplex hack, stable for all of the large projects. Q26 up, for those wishing to push the envelope. Q40 for pre-assembly polished reads. And things that we have increased since London Calling is consensus and variant calling with Medaka and CLAIRE 3. And these are things that we will continue to drive to improve the system. And all of this is delivering complete rich genomes with end-to-end protocols on single PromethION flow cells, and you're getting the modifications, the SV calling, the phasing, the SNV calling, and the many other things that all of you are exploring and talking about over the next day or so. It's really wonderful to have tools like this, but you've got to be able to use them. And so not everyone is expert in command line, so our epitome team have been working tirelessly to keep making bioinformatics more and more usable, more and more approachable. So our epitome team's got fantastic user interface, it's got a lot of pre-configured workflows, a lot of the Medaka improvements that you've just seen are going to be inside epitome really, really soon, so you don't need to go into the command line if you don't feel comfortable there. One of the big things that we've asked of the team to do is to really make the whole platform seamless. And what we had before was an old sort of legacy cloud, and it wasn't quite interacting with all of the work that the labs team were doing on the desktop applications. So we will be discontinuing and bringing down the old legacy cloud platform. Everyone who's a customer should be having emails from the teams about this. It becomes read-only from the 1st of October. And the new Epitome Cloud is live today. And so you can see here from the interface that we've put up here, it's completely seamless. You're either running local, you're running cloud, all of the same workflows all packaged up, really, really easy to use. So now that we've got the back-end bioinformatics getting easier and easier to work with, it's really important to pair that up with all of our protocols. So we have over 80 protocols in our community. We've got loads of different data analysis pipelines as well. And so what we've started to do is combine these end-to-end workflows from the lab to the analysis. Very importantly, we've just done a really big refresh on our documentation center. It is public. You do not need to log in. And you can go there to see all of the workflows that the team are working on. On the synthetic biology side, this is a lot of the work that's driving that really big interest in from the biopharma teams, but also anyone who makes and manipulates proteins. Everyone needs to be able to sequence plasmids, sequence amplicons, explore that you've got the right material, avoid months of failed experiments through not doing adequate QC. On the end-to-end workflows for small genomes, we've got, of course, sort of 16S in all of our SARS-CoV-2 genome, SARS-CoV-2, we've forgotten how to say it. It's only been 2 years. We've got all of our COVID workflows. But a new one that we've launched recently is direct from colony. So this is a really simple workflow for anyone working with isolates. And of course, the NOMIS, the nanopore-only microbial isolate sequencing workflow that we launched earlier this year, complete, gets you fantastic assemblies. You saw the results just earlier from the team. Q60 assemblies, perfect bacterial samples, really rapid, easy to use, and ready at the click of a button in Epitome. On the human side, we've got a beta going on right now for rapid whole genome sequencing for NICU-PICU applications. The cell-free DNA, which is a really fascinating area to look at if you want to start getting into fragmentomics, tumor normal, single cell, taking your single cell experiments to new lengths. And of course, we have the human variation workflow that captures all of your modifications and your variants. All of these workflows are going to get productized into Elysian. I've seen, again, it's been a very popular attendance of the show. Lots of questions going in. So Elysian is a fully automated sample-to-answer sequencing device. It right now has 3 MinINEs. The one outside has got space for 3 MinINEs on it, but it will also have a P2 solo and is a complete hands-off, fully flexible, full sample-to-answer product. So now that we've packaged all of these workflows, there's lots of them. There are a couple that I really wanted to go into in a little bit more detail, and these are around the human workflows. We've had fantastic talks today around sort of the human genomics. You've had the sort of the clinical breakout sessions, and what we're seeing a lot is an increased demand for how do you work with long reads in large cohorts. So there are always a couple of starting points. Number one, you need to create your own reference. So that's with the Nanopore only T2T. Step 2, you need to sequence an enormous number of genomes in order to get all of that wealth of information with a highly scaled production workflow. So Nanopore TDT, we spoke about at London Calling, but we'll do a refresh here. For Nanopore TDT, we use our ultra-long chemistry, generating incredibly long reads. And with that, you get Q40 assemblies. You get about sort of 20 large graph components out of running 2 flow cells of ultra-long. What we do then is we polish that with our 6B4 chemistry. So we add 1 flow cell. That takes your consensus accuracy up to Q50. And then the final thing that we do is we add POR-C data. And that then yields 30 TDT contigs, which is state-of-the-art, all on one single platform. The workflow is in Dorado, the assembly is in Dorado, and the entire package of flow cells and kits is available in the shop and shipping now. So all you have to do is scan that link, and you'll be added to the TDT shop. So the other part that you have to do then is how do you sequence thousands and thousands of genomes with Nanopore to get into all of the insights. So this is where we started, and we've done, with this type of workflow, we've already done over 100,000 genomes with certain partners. So our partners start with their samples. There's automated extraction, plate-based shearing, automated prep, plate-based QC. They load a sample per flow cell. They insert the flow cells into their sequencing units. They pause it. After 24 hours, they reload the flow cell and they carry on sequencing. Really important things that we've been doing on the back end is ensuring that we have keep-up base calling on the PromethION. That's all in place now. We're creating BAM files. So when you're in production, no one's keeping the fast fives or pod fives. They're all working in BAMs. Those BAM files get uploaded into Epitome and potentially into tertiary analysis providers. When running like this, customers are typically getting over 90% of their runs over 100 gigs. So the question is, in production, you want over 99% of your genomes getting to your target coverage. So one of the workflows we've been developing is a high-throughput barcoding workflow. So you take 96 barcoded libraries, you pool them, you sequence them so that you can balance the different barcodes, you understand the abundance of each sample. You then pool into 4 lots of 24 samples by abundance, and you load those into your sequencers. Now, some customers are carrying on with wanting to get the depth, a higher depth genome, and so they're putting 24 samples across 24 flow cells. But if you want to start getting into a lower, potentially a lower price, you could put 24 samples on fewer than 24 flow cells. And what the team have shown internally is that if you ran 24 samples, you may have a flow cell that doesn't quite get you your 30x coverage. However, if you pool them, multiplex them, and run them across 24 flow cells, all of your samples now reach 30x for true production-grade sequencing. This is a slide that you may have seen many times from Nanopore, which is sort of showing you our progression towards a $200 genome. And what this method really unlocks is our ability to actually start getting to a lower genome price sooner. It's also going to be the method when we start getting towards even higher and higher outputs. It's also going to be the method that actually delivers and enables that. So the final workflow that we've ended up with, alongside all of the work that we've done with our partners, is 96 samples. Of course, you're barcoding now, so LIMS becomes an essential tool. All of the automated sample prep workflow. You're loading your samples across your 4 sequencers. You're running, you're doing a pause, you're reloading 24 hours later, and you're carrying on. And everything is completely LIMS tracked. Now, there are a couple of things that we still think we could do better, but for that, I will let Lakmal come on stage and tell you about what he's got planned for the next few months. Thank you.
Thank you, Rosemary. I don't know why we thought like spinning the pixies is a good thing to do, but it's anyway. So as Rosemary said, so she talked about what we can currently do, what we currently offer, and I'm going to talk to you a bit more about the next frontier and what we are going to come up with in the next few months to years. So, so Rosemary mentioned that we are looking down our products and chemistry for applied markers, which is the right thing to do. And we have quite a lot of customers, and especially biopharma, like requesting us to look down our chemistry and platforms. And we are making our products and platforms and chemistry a lot more stable. And when we look down our products, but we still, our R&D teams are continuing to innovate, and they are coming up with new pores, new enzyme, new sequencing methods, and so on. So we will continue to innovate and come up with a new innovations. So some of the things I'm going to talk to you about are in the very early stage, in the almost like larva stage of this life cycle. Some of the things probably are in the middle, and some of the things, butterfly, are probably about to fly away. So when we improve our platforms and our chemistry, there are like multiple components that we need to tackle with. So we have nanopores, we have enzymes, and we have run conditions, sample preparations, pH, temperature, and so on. Then we have platforms and hardware, which all combinedly give a nice signal. And then we have our software architecture, training data sets, base caller, which converts the signal into the sequence. So when we improve our platforms, our accuracy or output, even though we individually, our teams individually improve each of these components, they all have to come together, and it is a combined effect that ultimately give you the upgrade that you require. So output is one of the key things that we are trying to increase at the moment. As Rosemary said, like for population genetic projects, we really need to cut our costs down, and one of the way to do this is improving the output. So at London Calling, we talked about a new buffer conditions, which enable us to eject blocked reads more efficiently, which ultimately gives us a better output, especially when the read lengths are longer. So this is also very compatible across our Kit 14 range and currently available flow cells. So it will be a seamless change to our customers. And more importantly, so we can improve the output either by reducing, so we can improve the output, we can get the same output, but with reduced number of washers and reloads, or you can get higher output if you do the same number of washers and reloads. So in this particular example, so you can see that we have few versions of this chemistry. Some are with developers, some are still in R&D. The one that are with developers, we have still, we have reduced the number of reloads or washings by 1. Instead of 3, we can do 2, but still you can get 15% more output. And in another version that we have in R&D, we can reduce the number of washings by 1, but we still can get 33% output. So this is a significant improvement, and all these improvements to output not only improves output for like T2T, but across the applications. And we have about 10 developers testing this new flow cell chemistry at the moment, and one of them is Matt Luce, from University of Nottingham. So he has used ultra-long kit to test these new flow cells. So as you can see here, so his N50 is 110 kb human, and he divided the sample, same sample, and then into 2, and used one of the old flow cells and one of the new flow cells, and with 2 washings, he got 34% more output compared to our old chemistry. So this is a significant improvement to the output, and we are very excited. He is very excited, and he did a long tweet today. And we are going to see what our other developers are coming up with in the next few weeks and months, but we are going to improve this even further, and we are going to improve the output significantly. So this is another improvement that we talked about at London Calling, improving enzyme motor speed to improve the output. So we have used machine learning methods to machine learning tools to improve our enzyme engineering. We have done quite a lot of work to improve the speed as well as the accuracy, and coupled with new enzyme purification methods, in high throughput purification methods, we have managed to make all these hundreds of different variants. We tested them, and then you can see in this plot, above the horizontal level, all the mutants are with higher accuracy, and towards the right side of the vertical line, we have number of variants that have higher speed. So the right top corner, you have mutants with higher accuracy and higher speed. So even though on the right-hand side graph that you can see that some of these mutants are actually running at high speed, currently our E8.2 runs at 400 bases per second, and some of these mutants, as you can see, runs at 800 bases per second. But we never managed to get the high output expecting from these faster mutants, mainly because when you run faster, you block pores more. So you can see from this graph that at early stages, it gives better output, but it plateaus out very quickly, mainly because when you run faster, more strands going through the pore, which means you block the pores more. So with these new buffer conditions, we seem to unlock that problem we had, and we are now getting quite nice higher output if we combine these faster motors with high output, faster motors with new buffer condition flow cells. So you can see here in this plot that we have got 270 GB output from a single Promethean flow cell. And I must say that this is in very early stages. This is probably the larva stage of the butterfly life cycle. But still, there's a lot more work to do, but we are very excited. You can see that output is quite nice. So this will enable us to do quite a lot of things, especially in T2T nanopore only assembly. As Rosemary said, at the moment we are using 4 flow cells, 2 for ultra-long, 1 for POSI, 1 for 6B4. And with this new improvement, we can reduce probably the ultra-long flow cell requirement from 2 to 1. So we are working tirelessly in R&D to reduce the flow cell requirement probably to 1 to 2 flow cells altogether. We are working on improving indels. Rosemary talked about improving MEDACA to improve indels. We are also working on longer read head pores to improve homopolymer accuracy as well. And then we are working on very long DNA sequencing that will hopefully get rid of POSI as well. So we have a workflow that we are, so our goal for a nanopore only T2T is to ultimately get to 1 or 2 flow cell requirement instead of 4, and this output improvement will significantly help that effort. So another significantly very important feature of nanopore sequencing is adaptive sampling. So many of you are using adaptive sampling, but for a long time we could not use 24 flow cells in one Promethean with our adaptive sampling. But with now like improvements to scripts and GPU configurations, we have unlocked that problem, and you can now do 24 flow cells in one Promethean with adaptive sampling, with also like 20% increase to sampling power. And in 6.2 Minnow, there's a new feature coming, like you'll be able to base call on target only data, enabling live base calling while sequencing. Previously, when you do live base calling, it compromised enrichment, but with the 6.2, you should be able to do this quite nicely without affecting the sampling power. Another new feature that we are trying to introduce is our R&D team is quite actively working on this, called sample until, or barcode balancing and target balancing method, with which we ask adaptive sampling to eject reads when a particular barcode reach a particular predefined threshold. So in this particular example, we have three samples barcoded: green, blue, and red. If you don't intervene, what you will get is what you see from these dotted lines. So the green will reach the predefined target in about like 15 to 20 hours, and blue will reach the target in about 35 hours, and red, as you can see, might not reach the target at all. So if you do this new barcode and adaptive barcode and target balancing approach, so when the green reaches the target, you switch off that particular barcode, and that will give a boost to blue and red. So as you can see, then blue goes to the target quickly. Instead of 35 hours, it will reach the target by 30 hours, and that will give another boost to red, and that will reach the target in 30 or so hours. If you didn't do anything, it will not reach the target at all. So this is a quite nice feature that we are trying to introduce, and hopefully all the adaptive sampling enthusiasts will get really nice data from this approach. So direct RNA is one of my favorites, and we have done quite a lot of work on direct RNA in the last year or so. So when we launched our DNA sequencing in 2013, it took like few years for us to release RNA kit. We did it in 2017, RNA001, and then RNA002 followed by quite quickly. But since then, we never really improved much because we had to do quite a lot of work on DNA sequencing. But last year, we released RNA004, which is now giving quite a lot of improvements over RNA002. Since we launched RNA004 last year, we have improved the method even more. So we have improved, we have introduced a new reverse transcriptase enzyme, which reduced the prep time by 20 minutes. At the same time, what it did was it increased the read length of the RNA. So in the middle plot, you can see old versus new. For both poly-A RNA and total RNA, the new method will capture longer reads compared to the old one, and that gives a significant boost to output. As you can see on the right-hand side, it's about like 20% output, like when it comes to 72 hours or so. We also enable short RNA sequencing mode. So this is now our default method. Previously, when you did RNA sequencing, you only managed to get reads above 200 bases, 200 nucleotides. But now we have improved our scripts and software, and now our default is reads longer than 50 nucleotides. And this short read mode enables people to do degraded RNA samples or shredded RNA samples, and more importantly, tRNA as well. So tRNA is becoming an important part of RNA biology. And you can see on the plot in the middle in this particular example, so we have RNA with N50, about 90 nucleotides, and you can see on the right-hand plot that you can see a significant boost to output if you have short RNA sequencing over the baseline where it used to be. So we are also trying to do more work on tRNA, but even with available chemistry, a lot of collaborators are working on tRNA, and you can see some of the tweets already. And we will work to improve tRNA offering even further. So accuracy on direct RNA sequencing improved significantly since we last talked about it at London Calling. Over the last year or so, it improved by about just roughly like 5 Q point since NCM 23. Currently, it stands at 19.4 Q, a 0.4 Q improvement since London Calling. And we are working on to improve it even further, and hopefully we will push it above Q20 soon. And our teams are actively working on this. So like in DNA, RNA modifications is important. So quite a lot of groups are using our RNA sequencing to do direct RNA modifications. So as you know, like Nanopore Technology is the only technology available to do direct RNA. And modification is one part of the one thing that you can't do with anything else. So we have released M6A and pseudouridine already. We talked about this in London Calling. So we have all context models with high accuracy. With M6A, you get 95.62, and with pseudouridine, you get 96. You can see in these plots, in the plots that IVG plots that you can call them very nicely. So we have recently included 2 additional modifications. This is Inosine and M5C. So Inosine and M5C are the next 2 most abundant modifications in RNA biology. So a lot of people were asking us to include Inosine in our pipeline. So with the latest so these are now actually available in Dorado, so you can do all 4 modifications at the same time. So this is, so you can see with just one click of a button, you can get all 4 modifications in RNA, Inosine, M5C, M6A, and pseudouridine in all contexts. And this is a very nice addition to RNA sequencing space. So as I said, we want to explore the full RNA biology, not only mRNA and tRNA, but ribosomal RNA as well. So this is this data is courtesy of Mithane Jain from Northeastern University. So in this example, you can see ribosomal RNA, how much modifications they have, and you can see quite nicely in the zoom-in version that we can call pseudouridine M56 very nicely. And this is a really good addition to our RNA sequencing. So quite a lot of people were asking about multiplexing RNA as well. So we are working in development to release this 24-plex direct RNA barcoding kit. So in the plot at the bottom, you can see there are 24 barcodes. To avoid variation introducing by different sample ligation efficiency, we have used the same sample with 24 barcodes, and you can see the barcode balancing is quite nice. So 24 barcodes means you should get about 4% coverage. And you can see all the 24 barcodes are quite nicely balanced. And on the right-hand side, you can see the classification accuracy, and they are quite nice as well, very high. Misclassification is quite low. And in the output plot, you can see the 24 barcodes are ligated to 24 different transcripts, and they are mixed together and run on a Promethean and Min9 flow cell, and output is quite high. So we are working still, we need to work on when it is poly A and total RNA, we are improving the output. Once that is done, I think the barcoding kit will be available for our developers to test, and hopefully we will release it soon. So there are quite a lot of happy RNA customers. You can see Alexander Wittenberg, I mean, they get Q21 accuracy, and Celgene tweeting about getting accuracy above Q20 as well. And Martin Smith, they are using direct RNA sequencing for long non-coding RNA sequencing, which you can't do with you can't do that with anything else. So there are quite a lot of happy customers in general RNA sequencing, but we are also working with biopharma, specifically with RNA vaccine manufacturing companies, and for them, two important features are poly-A tail length estimation and pseudouridine model, pseudouridine calling. So you can see on the plot on the left-hand side that each dark line is where the poly-A tail length should be, and each transcripts you can see that we can call very nicely, and they are ranging from 10 to 150 or so. And within that range, we can call the poly-A tail length very nicely. And M1 pseudouridine modification is critical for stability of mRNA vaccines, and we can call that quite accurately, around 97.2%. And these two features quite useful for mRNA vaccine manufacturing, biopharma industry. So for them, there are three tests: so identity, integrity, and purity that they need to get done. And under each, there are critical quality attributes that they need to tick off before they release mRNA vaccine batch. With currently available methods, they need to use multitude of different technologies, different equipments, expensive, and human resources high, and it takes weeks and months. But with direct RNA sequencing, all that can be replaced with one experiment. And you can see that sequencing confirmation, modified base caller, intact transcripts we can already do with poly-A tail, and we are working on 5' cap detection and double-stranded RNA, and that can be nicely coupled with our Q-line of GridION to do plasmid precursor test as well. So this is a quite useful package for biopharma manufacturing, and this is actually now available with commercial agreement for biopharma industry. So in summary for DNA, summary for RNA, so we can now do RNA sequencing at 98.9% accuracy, and we can capture longer transcripts with the new improvements to the protocol. We can do short RNA sequencing, anything above 15 nucleotides will be read. And we can do 4 modifications: M6A, 5MC, pseudouridine, and inosine. And we do have mRNA QC version 1 ready for biomanufacturing. We are working on multiplexing in R&D, target RNA sequencing. We are also working on automation, which is quite important, sample prep updates, and we are working on, obviously, to add more modifications, and also on a tRNA base calling model. So this is our roadmap. So Rosemary talked about what we offer now, and some of these things are already ticked, from hardware to platform to software and algorithms and to end-to-end workflows, what we can offer in 2024 and what we will offer in 2025. Obviously, there are quite a lot to do. So beyond DNA and RNA, so I have one more slide. So as you know, like, our platform is very versatile. It's very modular. Without redesigning the entire system, we can change the platform from one application to another by just changing the pore or just changing the chemistry. So we have designed it that way by design, which allows us to introduce new applications. So Gordon talked about multiomics in the morning in his talk, and proteomics is a big part of multiomics. So we talked about protein sequencing in the past, but we haven't said anything since then. So over the last couple of years or so, like our teams working very hard to improve protein sequencing technology, and they have done quite a lot of amazing work. There's amazing progress, but we still have more to do. So I'm not going to give an update on protein sequencing today, but I want to highlight this paper that came out last week in Nature. So this is from Jeff Nivala from University of Washington, and he's one of our very close collaborators. So working with us, he did this proof of concept of sequencing long protein strands with single amino acid sensitivity on a MinION on R9 flow cells. So as I said, just by changing the pore, you can adopt new applications. So in this particular example, in this method, so he used a new motor, a protein motor called ClipX. Like we do in DNA sequencing, we use a motor to pull the DNA out. And in this case, he used a ClipX to process the DNA, process the polypeptide through the nanopore, and by doing so, he can get a single amino acid sensitivity. So in the graph on the top right, so in this particular example, there are about like 60 amino acids, and in the middle, he changed the amino acid to individual 20 different amino acids, and by depending on the volume and the charge, you can see you can clearly identify the amino acid, or it gives a different signal based on the volume and the charge in the signal space. So that gives the signal a single amino acid sensitivity to detect amino acids in a polypeptide. Like we do in DNA, RNA with modification, we can also call post-translational modifications in peptides. So the graph at the bottom, so he used a kinase enzyme to print phosphate groups along the polypeptide in different ratios. So as you can see, depending on how much phosphate is there, you can see different signals, which clearly shows that post-translational modifications can be detected through nanopore. So this is quite a nice proof of concept study to show that we should be able to read polypeptides through nanopore on Oxford Nanopore platform. So we are quite excited to see this paper, and if you want to know more about protein sequencing, do come to London Calling next year. Thank you.